areas or all around the globe. And thank you so much, Kabir, for this invitation. It's also an honor to be here. So we're gonna start talking about this amazing um, field of knowledge, which is immunology, and especially talking about the complexity of autoimmunity. It's just gonna be a brief review of some molecular mechanisms which are described to understand a little bit of the pathophysiology in some of these autoimmune diseases. Well, we're going to start talking about what's the immune tolerance and how this balance is disrupted, because this is the basis of to understand especially the autoimmunity. It's been a century since in 1901 the physician Paul Ehrlich first speculated that the immune system might target host tissues and, and try to attack them as strange. And he coined his now famous term, the horror autotoxicus. I believe that Ehrlich never could imagine what we know right now in this moment about this fear that he had about the horror autotoxicus, and that now we describe and we know as autoimmunity. So I'm going to start talking about how important are autoimmune diseases in the clinical field. So why are they a significant clinical problem? First, autoimmune diseases have a chronic and degenerative nature. This leads that patients with these diseases, they never, as some other infections or other treatments, they never recover at 100%, and they live in the rest of their lives with this condition. So it's still a problem because it's in consequence they are associated with a high healthcare cost, and the treatment that these patients receive, it's a long-lived treatment. So in the field of the research and the clinical immunology and the rheumatology, there are a lot of research and studies that they continue to try to study the so many mechanisms and new molecules that could target and could stop at least the chronic inflammation, which is a key feature of these autoimmune diseases. Also, some of these autoimmune diseases have a high prevalence, especially in young populations during the prime of their working and the peak reproductive years. We can see this, for example, in the systemic lupus erythematosus, where young women develop the clinical symptoms at an early age, and this intermittent appearance of the clinical symptoms, they may affect obviously their daily activities, and could affect also their work and the quality of life. Or the most of the current therapeutic agents target the terminal phase of the inflammation. We're going to talk a little bit about this the important role of the chronic inflammation in the development of the autoimmune diseases, but this is a problem because they just try to make this inflammation, they try to reduce the inflammation, and they not stop, for example, the initial factors that trigger the autoimmune uh, complex network, that especially it's a multifactorial condition. So this is a problem, and we cannot revert, or we cannot, for example, prevent these autoimmune diseases. And finally, some diseases are limited to particular tissues, but some others are systemic or disseminated. We have so many different conditions. For example, type 1 diabetes, we have autoimmune thyroiditis, we have rheumatoid arthritis, we have lupus, we have Sjogren's syndrome. And this broad spectrum of the clinical disorders that the patients experiment, It's something that for the clinical diagnosis or criteria classification, they're constantly changing, and it's kind of difficult for the clinicians to generate a consensus. And because of the broad spectrum of the manifestations that it may appear in this kind of patients. So to start talking about the autoimmunity, we have to review briefly the mechanisms of the immune tolerance. What is tolerance? This is a state of unresponsiveness in which the lymphocytes remain alive but cannot exert effector functions against a particular antigen. We know that the main organ of the T cell tolerance is the thymus. So we know that all the lymphoid cells, they generate from the bone marrow, and especially the T cells, they migrate to the thymus, and in this tissue, they experience some mechanisms known as positive or negative control mechanisms, that these cells are continuously challenged by the epithelial cells in the cortex and in the medulla, especially in the medular thymic tissue, and those cells, which especially they don't recognize the self antigens, and in the other hand, the cells that they had a strong response, they are deleted, and this negative selection, which occurs in the thymus, is a process called the central tolerance. Those cells that have a well response, they could continue to their development stages, and this is called the positive selection. Also, when self-reactive T cells escape into the periphery, there are some other mechanisms which are known as peripheral tolerance that ensure that they are deleted or may become anergic. Energy state is a functionally unresponsiveness to the antigen. So these mechanisms also contribute to have, if you know, it's kind of a quality control that our immune system makes to assess that all of those cells that are circulating through the periphery, they are also functional, but they have to react what they are supposed to react, not just especially those of the pathogens as bacteria, viruses, fungi, and not to some of the antigens that we, especially in the proteins of our tissues, we produce or we express. So this is an important feature of the immune system, because if we don't have this immune tolerance, as we may know, the things is gonna get out of control. As you may see in this graphic, especially during their development in the thymus, these thymocytes or T cells that are known especially in the thymus, with high self-reactivity are deleted and removed, especially from the T cell repertoire. They enter these cells uh, experience the apoptosis, and as you may see, especially this group or this specific T cells clones are the one that they're gonna continue to their positive selection. But if they react, especially with a high affinity for the self MHC, this pool of autoreactive cells could be generated, and if they don't this, they don't get eliminated, they may escape the mechanisms of central tolerance, and this is a risk that for the immune system that we don't want to we don't want them to appear in this context. So the fundamental questions about autoimmunity are how tolerance to self may fail, and how these autoreactive lymphocytes are also activated, and they contribute to activate all of the immune responses that we know. especially not only in the T cells, we have some other mechanisms that occur also in B cells. And there are so many questions around this system and around this complexity that we are still researching, and it's a complete field of study. And some of the hypotheses that could or especially that are well studied, they are the genetic mutations, especially in some of the genes that are involved in the mechanisms of central tolerance. For example, we have the expression of the polymorphisms in the IRE gene. This IRE, or autoimmunity regulator, it's a gene that, especially the medullary thymic epithelial cells, they express, and they're continuously generating self antigens, and they present these antigens through the MHC complex to these T cells to be recognized, and especially to make these checkpoints that they elicit the mechanisms of the negative or the positive control. So obviously, if we have some mutations or deletions
Yes,
in this context, it's
para
uh,
afuera.
it's gonna
A mover
result
el paso, ahorita
especially
mover el paso para allá afuera.
fa
No hace
in a
que llegue
consequence
una parte de nosotros.
of the loss of tolerance, especially. So if you may see, this is the normal mechanism that may occur. So this is the sorry, this is the positive selection T cell which is developed, and they, for example, they they they're successful to this test, and this may through the periphery. Some other mechanisms that also occur in the thymus is the generation of the T regulating cells, or Tregs, and these cells are also important, especially in the periphery, because these Treg cells, or T regulatory cells, they're gonna work, for example, as guardians, or for example, police officers, as I like to tell, that they control and they um, minimize, for example, the immune responses, which is also an important feature of the immune system, because when, for example, we have an infection, we know that we have to start these mechanisms to control or to stop the pathogenicity, and that is, for example, activated by a bacteria, or for example by viruses. And once this infection is eradicated, we have to get things back to normal. And these mechanisms are turned off, for example, by but some many mechanisms, but contribution is very important for Treg cells. These cells that you're watching here, this AT means the autoreactive cells. Some of these autoreactive cells that escape and go through the periphery, and for example, this is another mechanism that have the B cells. When they change the receptor, or they edit this receptor, and this contributes to have a B cell receptor, which it's may recognize some self antigens, we have this potential of developing these autoimmunity mechanisms. There are some factors, or there are some several mechanisms that could trigger this autoimmunity. I'm telling you, this is not in some patients, for example, in some studies, we have very controversial results, because in some patients we have, for example, a high gene responsibility, for example, and but or they have this predisposing genetics that may affect or contributes to develop faster these mechanisms, in some others, as we're gonna talk about, for example, the Sjögren syndrome. And the sex, for example, the sex bias is also important to some people, and attribute this effect, for example, for the hormones, or for example, many other ambiental factors such as infections, or the molecular mimicry, etc. So these factors that you're watching here in the red box, they contribute, and maybe they and together with multifactorial contribution may develop especially this autoimmunity. So autoimmune diseases, like many other complex disorders, are believed to arise from several combination of genetics and environmental factors. I talked recently for the ARIA gene, but there are some other polymorphism described in the HLA or the MHC regions, and some other, for example, involving the cytokine pathways. Uh, Talking about the environmental factors, there exists infections, or for example, right now we have the microbiome, which may also contribute to regulate or to turn on or turn off some of the immune responses, which is also something very interesting to study. Or, for example, we have a defective regulation in the mechanisms of the peripheral tolerance. Some of a sample hypothesis is that polymorphisms in various genes may result in this effective regulation or reduced threshold for the lymphocyte activation. And as we are going to talk right now in this slide, there are so many environmental factors that may contribute, and they are also studied. For example, the impact that we have for some drugs and some chemical substances that may lead, for example, or they could trigger, and so mainly the innate immune responses that trigger, for example, the interferon pathways, and they contribute to the expression of the cytokine, the interferon, especially the type 1 interferons, which is a very classic signature in most of these autoimmune diseases. We have, for example, the infectious agents contribution, especially with some viruses that are also described, that this recognition of the RNA, the viral RNA by the TLR receptors or some other cytosolic receptors may contribute also to this production of type 1 interferons. We have, for example, the cigarette smoking, which is very classic, and for example, in rheumatoid arthritis, which may contribute to the generation of reactive oxygen species, or for example, the contribution of the some biochemical modifications in some antigens that could also be recognized by the receptors of the innate immune responses, which promotes also the production of the pro-inflammatory cytokines, or the ultraviolet radiation, which contributes to the apoptosis and the destruction of the cells in some other tissues, may contribute to the autoantigen exposure. For example, those ribonucleoproteins proteins, which are localized, for example, in the nucleus, that contribute to some processes like DNA replication or transcription, they, for example, the peripheral cells, they don't know these proteins or these antigens because they are mainly most of the time in the intracellular location, and this also may contribute to the activation of B cells, the production of autoantibodies, which is a classic mark also in these autoimmune diseases. And this is, for example, a key factor for the diagnosis, that we may recognize these autoantibodies responses, and also by the contribution of the type 1 interference. This graphic shows here the some other contributions of these environmental factors that trigger the development of the autoimmune diseases. We had three examples. For example, in the subject 8, it's a non-susceptible individual, which means that his or her gene background, they don't have some polymorphisms of risk or some other involved in the HLA region, or some cytokine pathways. So if this person is continuously exposed to these environmental factors, it is possible that their immune system may control or regulate or not trigger this autoimmune response. But in the context of an individual with this genetically predisposition, and for example, with a very low incidence of these contact with the environmental factors, this may just try to start an immune response, but this may not clinically be significant. In the contrast, for example, with a subject or a patient that has the genetic predisposition and so many factors in their environment that could lead by several years later the manifestation of these signs or symptoms that could finish or in the manifestation of the clinical disease. So the in autoimmune diseases, the control, not just by not just the loss of the tolerance in these mechanisms in central or peripheral tolerance is important. The control of inflammation is also lost. This results in a continuous immune activation without any other infection. This is very heterogeneous, and in some conditions, for example, they have so many peaks of activation or regulation that may contribute to a chronic inflammatory state. There are two hypotheses that is or two models for studying this condition. First, we have a barrier control between the innate and the adaptive immunity that could be distorted. This may continue or promote the inflammation by a positive feed-forward loop or a positive retroalimentation, or that may consistent with the interferon effects. Or the second model of study or the hypothesis is that we have the impaired reactivity of adaptive immunity with some reactivation of these autoreactive cells that could result in the persistent inflammation and clear defects on the tolerance checkpoints. These two models or two hypotheses, they're not mutually excluded to themselves. I think also that these may contribute or they're complementary, and in the context of some many autoimmune diseases, we can tell that this is also true for both models. As you may see, this diagram shows what are those effects that could be developed when some individual, which as we're telling, it has a genetically susceptibility, and it's also combined with the environmental factors. So one of the classic signatures or marks of these patients is the exacerbated productions of type 1 interferons. This overproduction of type 1 interferons, it will have an effect, especially in some other of the immune cells. About 400 genes are stimulated by the activation of the interferon pathways. For example, in the monocytes, we have a higher differentiation to dendritic cells. In these dendritic cells, which we know they are very important in the antigen presentation, they stimulate the production of the MHC molecules. They contribute to the production of some cytokines, which are very important. As you may tell here, we have the B cell activating factor known as BAFF that we're going to talk about it later. And they express more costimulatory molecules on the cell surface, and they also contribute to the T cell activating capacity. We have, for in the other hand, the natural killer cells or some other non-hematopoietic cells that contribute also to the expression of the chemokines or cytokines that this may contribute to the positive or the continuous loop that is classic in the autoimmune diseases. We're gonna focus right now in the role that T cells and B cells may contribute, especially the T cells for in the activation and the contribution or coordinating all these immune responses, because this is also very interesting that these positive loops they don't they're really good coordinated, and the organization that we can tell, for example, in some tissues where we have an inflammatory infiltration, they also occur just very similar to the normal, or as we can tell, in the healthy secondary lymphoid tissue. And the B cells, of course, they contribute to the production of the autoantibodies, which is very important. So we have these both bidirectional uh, communications, or contributions of both sides of the immune responses. For one hand, we have the contribution of the innate system with the production of the cytokines that promote especially the activation, differentiation, perpetuation, and the production of autoantibodies in the adaptive immune system. But also these cells also contribute to promote the pro-inflammatory state by neutrophils, by monocyte macrophages, natural killer cells, etc. So this is just a review of this complex system, but we're gonna take the model of study of the Sjogren syndrome, which is a multi-organic autoimmune disease, and this is the main theme that we're gonna discuss about the article that we're going to talk about. So, what's primary Sjogren syndrome? Sjogren syndrome is an inflammatory chronic autoimmune disease characterized by the lymphocytic aggregates in exocrine glands. Nevertheless, this infiltration is not just gonna cause hypofunction of the salivary or the lacrimal glands, which is of course the key feature of this disease, but also it's gonna contribute to the systemic multi-organ manifestations, as you may see in this picture. Patients with primary Sjogren syndrome often present dry mouth, dry eyes, fatigue, pain, and they are also they have an increased risk for developing B-cell lymphoma. And as you may tell, this disease mainly affects the women all around the globe. We can tell in this graphic we have an analysis of several autoimmune conditions, and you can tell the incidence in Sjogren syndrome is really clear. This difference with female to male ratio of 9 to 1, especially in the middle age, in comparison with some other autoimmune conditions. This disease is generally diagnosed, for example, in the fifth decade of life, with a mean range ranging from 51.6 to 62 years. However, the first symptoms may occur years before diagnosis, and this is also a problem because some of these symptoms, they are not clinically relevant, for example, for the patients, and this is there are several cases which are subdiagnosed, and until we have a clinical manifestation which affects, for example, the nervous system, the renal, the kidneys, or the lungs, or for example, the fatigue or the articular manifestations are more evident. This is the moment when the patient goes to the clinical to get the clinical diagnosis by a specialist. So those cells and these immune network that we're talking right now, it's also present in the pathophysiology of the primary Sjogren syndrome. There are so many factors that contribute to this gland, especially in the epithelial cells in the glands, that contributes to the destruction or the apoptosis of these cells, and this may contribute to the exposure of the autoantigens. The cytokines, the pro-inflammatory cytokines, and the production of type 1 interferon contributes or leads to the activation of CD4 positive T cells. Of course, their differentiation into the different phenotypes, for example, Th1, Th17 cells, and of course, with the respective production of their signature cytokines, such as interleukin 6, interleukin 12, interleukin 23, interleukin interferon gamma, etc. That also it's gonna start to initiate a recruitment of these cells into the exocrine glands, and of course this inflammatory infiltration is gonna start to damage the whole tissue and leading, for example, to the gland hypofunction. But the interesting thing here is that it's not just T cells or inflammatory cells they are recruited to the glands. Also, B cells start coming to this immune local response, and the B cells have this potential of generate these germinal center like structures that is promoted, for example, by some cytokines, especially by BAF. And the important why is this important? Because these germinal center-like structures are very similar to those that we're watching in the secondary lymphoid tissues, and it's a sign that B cells they don't require it anymore to go or to get differentiated or activated in the other lymph nodes. They could make that over there just in the tissue and promote, for example, the generation of plasma cells and locally the autoantibody production. And this is really interesting for us, and this is the key feature of this is the background of the study that we developed. As you may see, there are several. This is just an illustration. This is the real microscopic images that we can use, for example, in the diagnosis or the classification of the patients with primary Sjogren's syndrome. That we have several stages or these histopathological conditions could help us to classify the cases where, as you can tell, this is a normal salivary gland with the acini or the salivary ducts that is are classic in this tissue. But in some patients that are experiencing the infiltration or the recruitment of these inflammatory cells, they could start as a non-specific chronic salidinitis, where we have a diffuse infiltration of these cells, but the classic or the criteria for classification of the primary Sjogren's syndrome is the development of this foci. This is a focal lymphocytic salidinitis that you can tell they have a very well structure, or there's enough number of B cells or T cells, we can tell just because of these H&E tension staining, sorry, but we can tell that the cytoarchitecture of the tissue is not like this anymore. And we have some other more severe cases with the sclerosing chronic salidinitis. So to in order to answer some of these questions and to contribute a little bit of the understanding of how these immune responses or how these immunopathogenesis occurs in the glands and of the patients with primary Sjogren's syndrome, we question that, or we hypothesize that the contribution of this cytokine known as B cell activating factor and the receptors could be really important and could be associated with these ectopical germinal center-like structures. So one of the key molecules that is also known or described by several years ago is this cytokine that is essential for the B cell survival, maturation, and differentiation. And this is made, for example, by the signaling pathways conducted by their receptors. BAF may interact with three receptors, which are BAF receptor or BAF-R, also known as BR3, that is involved especially in the contribution of the survival and maturation of the B cells at the early stages. We have also TACI, which is important in the antibody responses, especially in T cell independent responses. They contribute also as a regulation or regulator of this B cell activation, and may contribute to the class switch recombination, especially for the production of IgA. And we have a third receptor, which is known as BCMA, that is expressed mainly by the final stages of differentiation, and is mainly expressed by the plasma cells. So this is really important in these stages. So the aim of this study was to analyze the BAFF-R, TACI, and BCMA glandular expression in minor salivary glands of patients, especially with those that show the germinal center-like structures, and how these signs or what we were watching or studying in the tissue could be associated with the serum levels of BAFF and the clinical severity in this disease. So as you can tell in the results of this study, we started to analyze the serum levels of these of the cytokine in both groups, especially with the primary Sjogren's syndrome patients versus the healthy subject control group. So as you can tell, we found that we have higher levels of soluble BAFF in the periphery, and with the clinical significance in this between these two groups. So the next question was to how are these also will these levels of soluble BAFF will be the same in all the patients? So we stratified and we divide the patients according to the histopathological conditions that I showed you before, especially those patients that they develop a non-specific chronic sialadenitis, and those patients with the focal lymphocytic sialadenitis that they did not develop the germinal center-like structures, and those patients that did develop the germinal center-like structures, and versus the healthy subjects. For our surprise, we found that the highest levels of soluble BAFF were found, especially in those patients that did not develop the germinal center-like structures. So the next question was to assess and to evaluate how is this for example, this finding the same will be the same in the expression of the glandular tissue. So we made some immunohistochemical stainings. First, we characterized these patients by the CD21 marker, which is a follicular dendritic cell marker that could lead us to identify and classify these patients into the study groups that I talked to you before. So we had another adjective that was okay. So what we're watching or what we're telling is that we have several cases with the lymphocytic infiltration, but with these cells be more so like a T cell scenario or a B cell scenario. So how are the proportions of these cells in these several conditions? So we also identified the subpopulations of T, B, and plasma cells by their classical surface markers, as you may see, CD3 for T cells, CD20 for B cells, and CD 138 for the plasma cells. And we found that, for example, it was obviously just it was just a confirmation in the context of these cases that those that develop the germinal center-like structures, they have the higher expression of the higher proportions of the B cells in this within this tissue. But as you can tell, there are some other important infiltration of the T cells in both cases, and this tells us that it is important the interaction or the contribution that both cells make to themselves. For example, we know that T cells also contribute and secrete some or some cytokines that help, especially to the B cells to continue to their differentiation stages and the development of autoantibodies and vice versa. So this is what we found, especially with the cytokine and their binding receptors in the tissue. We found something very interesting, that is the expression or the patterns of expression that it may also could be compare within the second lymphoid tissue that we have previously described in another article. And these especially tell us that these these receptors, they don't express as a homogeneous pattern. uh, and for example, we have these BAFF-R, which is the most relevant discovery of this study, that, for example, in the germinal center-like structures, as you can tell, there's a pattern all around the germinal center, and this, for example, may correlate with the type of B cells that are expressing or are infiltrating over there, that they don't have the same phenotypic or phenotype, that those cells that initiate or they started the germinal center reaction, or the maturation, or for example, the hypersomatic mutations, or the class switching. So this could be some 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 relevant information to understand what's going on between these tissues. We can tell that TACI and BCMA, they are expressed with a lower expression, but they could be also important in these responses. So finally, we correlate these findings with some clinical manifestations of the patients, and we found, for example, that the disease duration, as you can tell in the first graph, is correlated with the higher um, soluble BAF concentrations. So this may be important because, as you can tell, as with more patients which are elder, they could contribute to this perpetuation, but the expression or the higher expression of the soluble BAF. And we correlate also the production of some autoantibodies, especially with the anti-La or SSB antibodies, and the anti-Rho or SSA antibodies in these patients, that are the clinical antibodies that are found in the patients, and make also, they are part of the clinical criteria for the classification of these patients. So this can tell us that, okay, so the perpetuation of these immune pathogenesis, or the infiltration that we're watching, especially within the tissue, could be also promoted by the progression of the disease in some of these cases. So I like to finish, especially with this model, or proposed model, of the BAF system role in the primary Sjogren's immunopathology. That, as you can tell, there is several findings that we could tell in this the results of this article. For example, in the non-specific chronic cellulitis cases, we have this low production of serum BAF levels, and also a diminished BAF glandular expression. But this scenario is promoted by, or the balance is more to the expression of the BCMA receptor, which it's also been described as a negative regulator. So this contributes to for example, a scenario where T cells and B cells are not out of control, for example. And in the other two clinical relevant cases with the focal lymphocytic cellulitis, we can tell that, especially in those who are not or they not develop the germinal center-like structures, we have these higher BAF serum levels and a average BAF glandular expression within the tissue. And it's important, it's promoted by the balance, in the other hand, by the higher expression of the BAF receptor. And especially in those patients with the germinal center-like structures, we have maybe not just a high BAF expression in the periphery, but it's more important the expression within the glandular BAF, that these cells contribute to express locally. So this could be maybe the difference that could explain the different contexts or the different consequences that these patients have, and this heterogeneous spectrum of those patients that have a higher or a severe clinical outcome of this primary Sjogren disease. So just to punctualize some conclusions, primary Sjogren syndrome displays a wide variety of clinical conditions where elevated VAF levels could be associated with a focal lymphocytic cell adeninitis, but also with the disease duration, with the production of autoantibodies, especially as we can tell in our study groups. Moreover, BAF and the BAF binding receptors expression differs from distinct features between these two groups of the germinal center negative and the germinal center positive groups. And the balance between the BAF binding receptors expression and the interactions of BAF with some other cytokines, such as, for example, some chemokines or some other interleukins in the local microenvironment, could be the key mechanism for the establishment of the germinal center-like structures while this disease progression occurs. In conclusion, this story supports previous evidence that considers that the active BAF system role is very important in the pathogenesis of the primary Sjogren syndrome and the need for the strong biomarkers in the disease, or especially the development of some other biological therapies that could be very important in the treatment of this patient. So thank you so much for joining this meeting, and also thank you for again for the invitation. If there are so many questions, we can have some minutes to answer this. Thank you so much.
Thank you, Frank, for this very interesting presentation. I have one question. We have several therapies for Sjogren syndrome, like topical treatments, glucocorticoids, or even biological therapies like rituximab. But is there any experimental therapy that can't prevent the development of the disease?
Right now, we don't have some therapies that could act, for example, at the start of the disease, because as I was telling, it's very difficult for the patients to realize how or at what time of their life this could trigger some clinical manifestations. So it's really hard to, for example, to know exactly the moment that could stop the development of these stages. Mostly the patients that go to the rheumatologist, they go with several clinical manifestations, such as, for example, with some obviously the dryness, but with the chronic fatigue, and there are some others that experience impaired renal function, for example, the cutaneous manifestations, or for example, neuropathies. So it's very difficult to revert these conditions, especially because the damage is very several. So the aim of the therapy in the treatment of these conditions, there are, for example, the corticosteroids that have a anti-inflammatory effect in these cells that are also, as I was telling, out of control. And at least we cannot revert, but at least we can stop the progression of the disease. So this is, but in the other hand, we have some several adverse effects that could also be not very well tolerated by the patients. For example, the affectation of the metabolism, the weight gaining, or for example, um, they have, for example, some other treatments that would be very very expensive for the treatment, like as you said, the rituximab, that could deplete the B cell populations. And also, I like to tell that the contribution of this work is, for example, some other evidence that we have another a biological treatment known as belimumab, which is an anti-BAF monoclonal antibody. So, but this drug or this monoclonal therapy is just approved by the FDA for the treatment of systemic lupus erythematosus, and it's not available still for the treatment of primary Sjogren syndrome. So the contribution of this research could provide more evidence that the role or the importance of this cytokine in the context of this disease that could lead to the treatment of patients with some more specific therapies such as belimumab. But we have, on the other hand, the high cost, and as we can tell here in our country, it's not accessible for all the patients to get to be treated with this biological therapy.
Thank you, Frank. Is there any other question?
Well, if you have so many questions later, you can find me right now. This is my email address, and we can still have in touch.
Okay. Well, thank you once again, Frank, for joining us today. And I'll let you know if some of the participants have questions. I guess there's no more questions. No.
Yeah,
No.
for sure. It's okay. It's okay. So thank you so much also for this opportunity. I'd like to say finally that to those who join us today as the very first time, this journal club has some interesting topics that they discuss. So keep in touch for more contents, and I hope that in the next months or weeks you can join to another meeting. Thank you so much.